Welcome, curious minds. Have you ever paused to wonder why you have your specific eye color, or maybe why certain family traits seem to, I don't know, skip a generation, only to pop up again later?
Yeah, for ages, scientists kind of scratched their heads over this. There was this idea, the blending hypothesis, right?
Right, that genetic stuff just mixed together like stirring paints. Seems logical on the surface.
It does, but it created more problems than it solved, really.
Yeah,
If traits just blended, how could something disappear and then reappear?
good point. And wouldn't everything just end up beige,
Yeah,
uniform?
exactly. That's where Gregor Mendel comes in. A monk working quietly in an abbey garden with, believe it or not, pea plants.
Humble beginnings for a huge idea.
Totally. His genius was proposing this particulate hypothesis, not blending paint, but passing on distinct separate units. We call them genes now.
Okay, so less like paint mixing, more like shuffling cards.
That's a great analogy. The genes keep their identity even when they're dealt out again in the next generation.
Got it. So today we're taking a deep dive into Mendel and the gene idea. We'll unpack how his, well, really meticulous experiments laid the groundwork for all of modern genetics, even before anyone knew about chromosomes.
Yeah, we'll break down his big laws, see how probability surprisingly plays a huge role, and then look at patterns that are actually more complex than what Mendel first saw.
And finally, connect it all back to us human traits, genetic disorders, things that affect our lives every day.
Right. Our mission here is really to pull out the most important stuff from this foundational science, help you grasp the core mechanisms and why they matter in the real world.
We want to make these tiny, complex processes easy to picture so you walk away feeling, really informed.
Let's do it.
Okay, let's unpack this. Mendel's approach was, well, just brilliantly scientific and quantitative. He chose garden peas, right?
Yeah, garden peas. They were perfect, actually.
Yeah.
Lots of distinct varieties. Think purple versus white flowers, round versus wrinkled seeds, things you could clearly see and count.
And crucially, he could control who mated with whom.
Exactly. So a character is something heritable, like the flower color. And a trait is the specific version, like purple or white.
Makes sense.
Peas normally self-fertilize pollen from a flower, fertilizes eggs in the same flower. But Mendel, he mastered cross-pollination.
How did he do that?
He'd carefully remove the pollen-producing parts, the stamens, from one plant, and then dust pollen from a different plant onto it. Very precise work.
So he knew the exact parents of every seed, no guesswork.
None. And he always started with true breeding plants.
Meaning?
Plants that, if they self-pollinated for generations, always produced offspring identical to themselves. Like true breeding purple always made purple.
Okay, got it. Then came the main event.
Then came hybridization. Crossing two different true breeding parents. He called them the P generation for a parental.
And their kids.
The F1 generation, the first filial generation.
But he didn't stop there, right? That was key.
Absolutely key. If he had only looked at the F1s, he would have missed the big picture. He let those F1 hybrids self-pollinate.
To get the F2 generation.
Right. And then he counted, meticulously, thousands of F2 plants. That careful counting led straight to his two fundamental principles.
Okay, so let's talk about that first cross. True breeding purple flowers crossed with true breeding white. What happened in F1?
This was the first big surprise. They were all purple.
All of them, not pale purple like blending would suggest.
Nope, all fully purple. The white trait seemed to have just vanished.
Ooh, okay. But then came the F2s.
Exactly. When those F1 purples self-pollinated, the white trait reappeared. And consistently in this ratio of about 3 purple plants to 1 white plant.
3 to 1. So the white factor wasn't gone, just hidden somehow.
Precisely. It wasn't destroyed or diluted, it was just masked. We now call the purple trait dominant and the white trait recessive, and this whole observation led to the law of segregation.
That 3.1 ratio is iconic. So how do we translate Mendel's heritable factors into modern terms? Genes, DNA.
It maps perfectly onto our modern understanding. Mendel's model basically has 4 parts.
Okay, lay them out for us.
1. Alternative versions of genes account for variations. We call these versions alleles. So for flower color, there's a purple allele and a white allele. They're just slightly different DNA sequences at a specific spot, a locus on a chromosome.
Got it. Alleles are different versions of a gene. What's 2?
2. For each character, an organism inherits 2 alleles, one from each parent. Remember, our cells are mostly diploid, 2 sets of chromosomes, so 2 alleles for each gene.
One from mom, one from dad.
Okay.
3.
3. If the 2 alleles are different, the dominant allele determines the organism's appearance, its phenotype. The recessive allele has no noticeable effect on the phenotype.
Ah, that's why the F1s were all purple. They had one purple and one white allele, but purple is dominant.
Exactly. And number 4 is the law of segregation itself.
Mm.
The two alleles for a character they segregate from each other during gamete formation, sperm and egg production.
So each egg or sperm cell gets only one of the two alleles the parent has.
Right. That separation happens during meiosis. That's the mechanism.
Wow. Okay, that explains the disappearing and reappearing traits and the 3.1 ratio. How do geneticists actually visualize this? Is there a tool?
There is the Punnett square. It's a really handy grid.
I think I remember those from biology class.
Probably. You list the possible alleles from one parent's gametes along the top and the other parent's along the side. Then you fill in the boxes to see all the possible combinations in the offspring.
So for that F1 cross, the self-pollination of PP plants.
Right. If P is purple and P is white, both parents make P and P gametes. The Punnett square shows you get 1 PP, 2 PP, and 1 PP combination.
Which means 3 genotypes give purple flowers, PP and the 2 PP, and one gives white PP. There's the 3.1 ratio.
Exactly. And that introduces some key terms. Homozygote means having 2 identical alleles like PP or PP.
Home meaning same.
Right. And heterozygote means having 2 different alleles like PP hetero meaning different.
Okay. And phenotype is what you see purple flowers. Genotype is the actual genetic makeup PP, PP, or PP.
Perfect. Now here's a practical problem Mendel faced. If you have a purple flowered pea plant, how do you know if its genotype is PP or PP? They look the same.
Right. You can't tell just by looking. So how do you figure out that genetic mystery?
You use a test cross. It's a clever diagnostic tool.
Okay, how does it work?
You cross your mystery purple plant with a plant, is homozygous recessive, a white-flowered PP plant in this case.
And the results tell you the answer.
They do. If all the offspring are purple, your mystery plant must have been PP because it could only pass on P alleles.
Ah, because P crossed with the PP would give some PP offspring?
Exactly.
Yeah.
If you get both purple and white offspring, roughly half and half actually, your mystery plant was heterozygous PP. It's like a genetic paternity test, but for peas.
Very cool. Okay, but Mendel didn't just stop with one trait, did he?
No way. He was thorough. He started looking at 2 characters at the same time, like seed color and seed shape.
Okay, what were the traits there?
Yellow seeds Y are dominant to green, and round seeds are dominant to wrinkled R.
Got it. So what did he cross?
He started by crossing true breeding yellow round plants, YYRR, with true breeding green wrinkled plants.
Yeah. Okay, so the F1 generation would get a Y from one parent, Y from the other, R from one, R from the other. It'd all be YRR.
Precisely. All dihybrids, heterozygous for both genes, and phenotypically, they all have yellow round seeds because Y and R are dominant.
Okay, now the big question. When these F1 dihybrids make gametes, do the Y alleles and the RR alleles stick together? Like, does Y always travel with R and Y with R, or are they independent?
That was the question. Are the genes linked, or do they assort independently? To find out, he did a dihybrid cross. He crossed 2 F1s, YRRXYRR.
And what happened? If they were linked, you'd expect just yellow, round, and green wrinkled offspring, right? In a 3.1 ratio again.
That's what you expect if they were dependent. But that's not what he found. He found 4 different phenotypes in the F2 generation.
4? What were they?
He found yellow round seeds, green round seeds, yellow wrinkled seeds, and green wrinkled seeds. And always in a consistent ratio.
Let me guess. It's not 3.1.
Nope. It was 9.3.3.1. 9 yellow round, 3 green round, 3 yellow wrinkled, and 1 green wrinkled.
Wow. That 9.3.3.1 ratio is famous too. What did that tell him?
It strongly supported the idea of independent assortment. It meant the alleles for seed color segregated into gametes independently of the alleles for seed shape.
So getting a Y allele didn't influence whether the gamete got an R or an R allele. They sort independently.
Exactly. This became his second law, the law of independent assortment. 2 or more genes assort independently during gamete formation.
That's another huge concept. Does it always apply?
Ah, good question. It applies specifically to genes located on different chromosomes or genes that are very far apart on the same chromosome.
Why does distance matter on the same chromosome?
Because of crossing over during meiosis. If genes are close together, they tend to be inherited together more often. But if they're far apart, crossing over makes them behave as if they're on different chromosomes.
Okay. So Mendel was maybe a bit lucky that the traits he picked happened to assort independently?
He was either lucky or incredibly insightful in his choices. It definitely simplified his results and let him see these fundamental patterns clearly.
It feels like genetics involves a lot of chance, like coin flips or dice rolls?
It absolutely does. Mendel's laws are basically reflecting fundamental rules of probability. The segregation of alleles into gametes, that's a 50-50 chance, like flipping a coin.
Heads or tails, P or P.
Exactly. And each segregation event is independent, just like coin flips. The outcome of one doesn't affect the next. This was revolutionary, applying probability to biology.
So how do these probability rules work in genetics? You mentioned multiplication.
Right, the multiplication rule. If you want to find the probability of two or more independent events happening together, you multiply their individual probabilities.
Like getting two heads in a row with a coin, that's half times half equals a quarter.
Perfect example. We use it in genetics all the time. What's the chance of getting an F2P plant with wrinkled seeds, genotype RR?
Well, if the parents are RR, the chance of getting an R gamete from the egg parent is 12. And from the sperm parent, also 12. So 12 times 12.
Equals 14. That's the multiplication rule in action.
Okay, but what if there's more than one way for an event to happen?
Then use the addition rule. If an event can occur in two or more mutually exclusive ways, you add their individual probabilities.
Mutually exclusive meaning they can't happen at the same time.
Exactly. For example, how do you get a heterozygous R offspring from an RRXR cross?
Well, the egg could be R and the sperm RR. The egg could be R and the sperm R. Those are the only two ways, right?
Right. And the probability of the first way, R from egg, R from sperm, is 12, 12 equals 14. The probability of second way, R from egg, R from sperm, is also 12, 12 equals 14.
So since those are the two mutually exclusive ways to get R, you add them. 14 plus 14.
Equals 12. So half the offspring are predicted to be heterozygous, the addition rule.
These rules seem really powerful. Can you use them for more complex crosses, like that dihybrid 9.3.3.1 thing, without drawing a huge Punnett square?
Absolutely. That's the beauty of it. A dihybrid cross is just two independent monohybrid crosses happening at the same time.
So you break it down.
You break it down. Consider the YYR-XYRR cross. What's the probability of getting YYRR offspring?
Okay, for the seed color gene, YYXYY, the chance of getting YY is 14. For the seed-shaped gene, RRXRR, the chance of getting RR is also 14.
Right. Since these are independent events because the genes assort independently, you multiply the probabilities. 14 for YY, 14 for RR equals 116.
So 1 out of 16 offspring should be YYR. That's much faster than drawing a 16-box Punnett square.
Way faster. You can calculate the probability for any genotype this way. And you can even extend it to trihybrid crosses or more complex situations. It's all about applying these basic probability rules.
And Mendel understood the statistical nature, which is why he counted so many plants.
Precisely. He knew that with chance events, you need large sample sizes for the observed results to get close to the predicted ratios. This is the law of large numbers. Brilliant work for his time.
Okay, so Mendel's laws are the foundation. But you mentioned inheritance can be more complex. It's not always simple dominant-recursive 3.1 ratios.
Far from it, often. Mendel picked traits that showed simple dominance, but nature is usually more nuanced. Most genes have more than two alleles, and the relationship between genotype and phenotype can be complicated.
But the core ideas still hold. Segregation, independent assortment.
Yes, those fundamental principles still apply at the level of how alleles are passed on. It's the expression of those alleles that gets more complex.
Okay, like how? What happens when dominance isn't complete?
Right, so we talked about complete dominance. PP and PP look the same. But sometimes you get incomplete dominance.
Incomplete, meaning the heterozygote is somewhere in between.
Exactly. Think snapdragon flowers, cross a true breeding red one, RR, the true breeding white one, WW, and the F1 hybrids, RW, are all pink.
So it looks like blending.
It looks like it, but it's not.
Yeah.
Because if you cross those pink F1s, RWX, RW, what do you get in the F2 generation?
Using a Punnett square, you'd get RR, RW, and WW.
In what ratio?
1 RR red, 2 RW pink, and 1 WW white. A 1.2.1 ratio.
Exactly. So the red and white alleles didn't blend away. They were both still there, unchanged, ready to produce red and white flowers again.
Yeah.
The heterozygote just has an intermediate phenotype.
Okay, that's incomplete dominance. What about codominance?
Codominance is when both alleles affect the phenotype in separate distinguishable ways. Not an intermediate blend, but both showing up.
Like stripes or spots?
Sort of. A classic example is the human MN blood group system. It's based on molecules on the surface of red blood cells. If you have the LM allele, you make M molecules. If you have LN, you make N molecules.
So what if you're heterozygous LMLN?
You make both M molecules and N molecules, not some intermediate molecule, but both distinct types are present. Both alleles are fully expressed. That's codominance.
It's interesting how the definition of dominance can shift depending on how you look at it.
It really can. Take Tay-Sachs disease, a tragic human genetic disorder. At the organism level, whether someone has the disease or not, the Tay-Sachs allele is recessive. Only homozygous individuals, TT, get the devastating disease. Heterozygotes, TT, are healthy.
So recessive at that level.
But look biochemically. The gene codes for an enzyme. Homozygous normal, TT, have full enzyme activity. Heterozygotes, TT, have about half the normal enzyme activity. Homozygous affected, TT, have virtually none.
So half activity, that sounds like incomplete dominance at the biochemical level.
Exactly. And now look at the molecular level, the protein molecules themselves. Heterozygotes produce equal numbers of normal enzyme molecules and non-functional enzyme molecules.
So both types are being made. That sounds like codominance at the molecular level.
Right. Same gene, same alleles.
Mm-hmm.
But whether you call it recessive, incompletely dominant, or codominant depends entirely on the phenotypic level you're examining. It's a great illustration of the nuance.
And it's also important to remember dominant doesn't mean better or more common, right?
Absolutely not. Polydactyly, having extra fingers or toes, is caused by a dominant allele. But it's quite rare in most populations. The recessive allele, for 5 digits, is far more common. Dominance just refers to the phenotypic effect in a heterozygote.
Okay, what about having more than just 2 allele options for a gene?
That's very common too. Mendel's peas had 2 alleles per gene, but many genes exist in multiple forms within a population. We call this multiple alleles.
Like the ABO blood groups in humans.
Perfect example. There are 3 main alleles, IA, IB, and I.
IA gives you A antigens, IB gives you B antigens, and I gives you no antigens.
Right, and these combine to create the 4 blood types. Type A can be IA, or IAI. Type B, IB, or IB. Notice the codominance here. And type O2. 3 alleles, 4 phenotypes.
Okay, another twist. Can one gene affect multiple traits? We usually think one gene, one trait.
That's often an oversimplification. Most genes actually have multiple phenotypic effects. This property is called pleiotropy.
Pleo, meaning many.
Yes. Think of genetic disorders like cystic fibrosis or sickle cell disease. A single gene defect causes a whole range of symptoms affecting different organs and systems.
Even Mendel's flower color gene did more than one thing.
It did. The same gene that determined purple versus white flowers also affected the color of the seed coat. One gene, multiple effects.
All right, now what about the opposite? Multiple genes affecting one trait.
Yes, that happens too in a couple of ways. One interesting interaction is epistasis.
Epistis, what does that mean?
It's when the phenotypic expression of a gene at one locus alters or masks the expression of a gene at a second, different locus.
Okay, one gene interfering with another. Can you give an example?
Labrador Retriever coat color is the classic example. One gene determines black, B, dominant, versus brown, B, recessive, pigment. Simple enough.
Black or chocolate labs?
Right. BUT.
Hmm.
There's a second gene, let's call it E, that controls whether any pigment gets deposited in the fur at all.
Ah, so it's like an on-off switch for pigment.
Pretty much. If a lab has 2 recessive E alleles, genotype E, it cannot deposit black or brown pigment in its fur, regardless of what the B gene says. The result?
A yellow lab.
Exactly. So the E gene is epistatic to the B gene. It overrides its effect.
What happens if you cross 2 dogs heterozygous for both genes, like BBX, BBE? They'd both be black labs, right?
They would.
Yeah.
But their offspring show a modified ratio because of epistasis. You don't get 9.3.3.1. Instead, you get 9 black labs, 3 chocolate labs, and 4 yellow labs.
9.3.4. That shows how 2 genes can interact to produce one trait coat color.
Precisely. Now, sometimes multiple genes contribute to a single trait in an additive way.
Additive, like adding up effects.
Yes, Mendel's traits were discrete, either were categories. But many traits, especially in humans, vary along a continuum. Think about height or skin color.
Right, there aren't just 2 height categories, tall and short. There's a whole range.
These are called quantitative characters, and they usually result from polygenic inheritance.
Poly meaning many genes.
Many genes influencing a single phenotype. For something like human skin color, several genes contribute to melanin production. Each dark skin allele might add a small unit of pigment.
So the more dark skin alleles you inherit across all those genes, the darker your skin?
Generally, yes. This additive effect of multiple genes creates that smooth spectrum of variation we see, often following a bell curve distribution in a population.
So genetics gets pretty complex beyond simple Mendelian traits. But is it only about genes? What about the environment?
An absolute critical point. Genes are not the whole story. The environment plays a huge role in shaping phenotype, nature and nurture.
Can you give an example?
Think of a tree. Leaves on the sunny side might be smaller and thicker than leaves on the shady side, even though they have the exact same genes.
Same genotype, different phenotype due to environment.
Exactly. Or for humans. Nutrition drastically affects height potential, exercise changes build, sun exposure darkens skin temporarily. These are environmental influences on phenotype.
Even identical twins with the same genes aren't truly identical phenotypically.
Right. Their unique experiences, diets, illnesses, all contribute to subtle and sometimes not so subtle differences.
So characters that are influenced by both genetics and environment are called
Multifactorial. Many factors, both genetic and environmental, contribute to the final outcome. Most complex traits, including many human diseases, are multifactorial.
Okay, let's shift focus specifically to humans. Studying human genetics must be harder than peas, right? No controlled crosses.
Definitely more challenging. We have long generation times, small family sizes, and of course, ethical considerations prevent experimental mating.
So how do geneticists study human inheritance?
One key tool is pedigree analysis.
Drawing family trees.
Essentially, yes. A pedigree is a family tree that diagrams the inheritance of a specific trait across multiple generations. Squares for males, circles for females, shaded symbols for individuals showing the trait.
And by analyzing the patterns, who has the trait, who doesn't, you can figure out how it's inherited.
Often, yes. You can deduce if a trait is likely dominant or recessive, and you can often figure out the genotypes of individuals. It helps predict the probability of future offspring inheriting the trait.
Like tracing something simple, maybe like widow's peak hairline.
Exactly, that's often dominant. Or maybe attached versus free earlobes,
Yeah.
or the ability to taste PTC paper, which is recessive. Pedigrees are crucial for understanding human genetic disorders too.
Let's talk about those disorders. Many are inherited recessively, right?
Yes, many genetic disorders follow simple recessive inheritance patterns. This includes things from relatively mild conditions like albinism to severe, life-threatening diseases like cystic fibrosis or Tay-Sachs.
What usually causes a recessive disorder at the molecular level?
Typically, the recessive allele codes for a malfunctioning protein, often an enzyme, or maybe no protein at all.
But heterozygotes who have one normal allele and one recessive allele are usually healthy.
They're called carriers. That one normal allele usually produces enough functional protein to prevent the disease phenotype.
So for a child to get a recessive disorder, both parents must carry the allele.
Generally, yes. Most individuals with recessive disorders are born to parents who are both carriers, heterozygotes, and phenotypically normal.
And in that carrier X carrier cross, like AXAA, what's the risk for each child?
Each child has a 14 chance of inheriting 2 recessive alleles, AA, and having the disorder, a 12 chance of being a carrier, and a 14 chance of being homozygous normal, AA.
That 1 in 4 risk applies to each pregnancy, right?
Absolutely. It's an independent event each time. The previous child's genotype doesn't change the odds for the next one.
Does the frequency of these disorders vary?
Significantly.
Yeah.
Due to different population histories, certain genetic disorders are much more common in specific ethnic groups, and mating between close relatives, consanguineous mating, increases the risk.
Why is that?
Because close relatives are more likely to share the same rare recessive alleles inherited from a common ancestor. So their offspring have a higher chance of being homozygous for those rare, potentially harmful alleles.
Let's look at cystic fibrosis again. You mentioned it's recessive and common in people of European descent. What does it do?
CF is caused by a defective chloride ion transport channel protein. This leads to abnormally thick, sticky mucus building up in various organs, especially the lungs, pancreas, and digestive tract.
Causing lung infections and trouble digesting food?
Yes. Chronic lung infections and poor nutrient absorption are major problems. It's a clear example of pleiotropy, one faulty gene, causing multiple symptoms throughout the body.
Now, sickle cell disease, you mentioned it earlier. It's a really interesting case, especially evolutionarily.
It really is. Most common in people of African ancestry, caused by a single amino acid change in hemoglobin, the protein in red blood cells that carries oxygen.
And this change makes red blood cells sickle-shaped under low oxygen.
Exactly. These sickled cells can clog small blood vessels, causing pain, organ damage, and anemia.
You said it acts differently depending on how you look at it, recessive, incompletely dominant, codominant.
Right. Organism-level disease. Recessive? Need 2 sickle alleles. Some symptoms under stress. Incompletely dominant. 1 allele is enough for trait. Molecular level. Codominant, both normal and sickle hemoglobin are made in carriers.
Carriers are said to have sickle cell trait. How common is it?
About 1 in 10 African Americans carry the allele. That frequency is unusually high for an allele that causes a serious disease in homozygotes.
Why so high? There must be an advantage, right?
There is, in certain environments. The key is malaria.
Malaria. How are they connected?
Individuals with sickle cell trait, the heterozygotes, have a significant resistance to the malaria parasite.
Wow. So carrying one copy of the sickle cell allele protects you from severe malaria?
Yes, particularly in young children. The parasite has a harder time surviving and reproducing inside those slightly altered red blood cells.
So, in areas where malaria is widespread, like parts of Africa, heterozygotes have a survival advantage over both homozygous normal individuals susceptible to malaria and homozygous sickle cell individuals have sickle cell disease.
Precisely. That's heterozygote advantage, a classic example of natural selection maintaining a harmful allele in a population because it offers a benefit in a specific environment. Genetics, evolution, and disease all intertwined.
Fascinating. Okay, not all genetic disorders are recessive. What about dominant ones?
Some are caused by dominant alleles. Achondroplasia, a common type of dwarfism, is one example. If you inherit just one copy of the dominant allele, you have the condition.
So most people are homozygous recessive for the normal allele.
Correct. Lethal dominant alleles are generally rare because if they cause death before reproductive age, they aren't passed on.
But there's a catch, isn't there? What if the symptoms show up later in life?
That's the loophole. If a lethal dominant allele doesn't manifest until after a person has had children, it can persist in the population.
And the prime example is?
Huntington's disease, a devastating progressive neurodegenerative disorder. It's caused by a dominant allele, but symptoms typically don't start until age 35 to 45 or even later.
So people can pass it on to their children before they even know they have it.
Exactly. And since it's dominant, each child of an affected parent has a 50% chance of inheriting the allele and eventually developing the disease. Genetic testing is available, but it presents a really difficult choice for people at risk.
Such complex ethical issues. Now, we've talked about these single-gene Mendelian disorders, but what about common things like heart disease or diabetes? They run in families, but not in simple patterns.
Right. Those are mostly multifactorial conditions.
Meaning genetics and environment.
Yes. They have a genetic component, often involving multiple genes, polygenic, plus significant environmental influences. Your lifestyle, diet, exercise, smoking, stress plays a huge role in your actual risk, interacting with your underlying genetic predisposition.
So your genes might load the gun, but environment pulls the trigger, so to speak.
That's a common way to put it, yeah. Understanding this interaction is key for prevention and treatment.
All this knowledge, from Mendel's peas to complex human diseases, leads to practical applications like genetic counseling, right?
Absolutely. Genetic counseling uses Mendelian principles, probability rules, pedigree analysis, and increasingly sophisticated genetic testing to help individuals and families understand their risk for inherited conditions.
So counselors help people interpret family history and test results.
Yes, they help prospective parents, for example, understand their chances of having a child with a specific genetic disorder based on their carrier status or family history. They might calculate the probability using the multiplication and addition rules we talked about.
Like, if two prospective parents find out they're both carriers for cystic fibrosis.
The counselor would explain that for each pregnancy, there's that 1 in 4 chance the child will have CF, 1 in 2 chance the child will be a carrier, and 1 in 4 chance the child will inherit 2 normal alleles. They provide information and support, but the decisions are up to the individuals.
And it's crucial to remember, again, each child is a new roll of the genetic dice.
Independent events.
Yeah.
Always.
Wow. We've covered a lot, from Mendel counting peas in his garden to the really complex web of human genetics and disease. It really shows the amazing architecture of inheritance.
It really does. These discrete units, genes, passed down according to predictable rules of probability, yet underpinning such incredible diversity and complexity.
Whether it's the simple elegance of a 3.1 ratio, the nuances of incomplete dominance in a snapdragon, the spectrum of human skin color, or that double-edged sword of the sickle cell trait.
Mendel's core ideas of segregation and independent assortment are still the bedrock. Understanding them gives us this incredible power to decode our own biology and the life all around us.
So next time you see a family resemblance, or maybe you're thinking about your own health history, remember that intricate dance of alleles happening behind the scenes. Those tiny packets of information have such a huge impact. What other traits do you notice out there that might have a cool genetic story? Makes you want to keep digging into that amazing deck of genes.
Thank you for joining us on this deep dive into the truly fascinating world of genetics. We hope you feel, a lot more informed now, and maybe even more curious about the genetic blueprint that makes every single one of us unique.